Hola, buenas tardes a todos. Good afternoon. I'm not Carmen Paradas. My name is Mónica Povedano. And Carmen, she can't be here today. So I am very happy to introduce the first session of this meeting today, because today is as you know, the ALS Day. And these two different talks are focused on ALS. I'd like to introduce the first speaker, Orla Hardiman. I have a good relationship with Orla. She is the head of Department of Neurology and Professor of Neurology in Ireland in the Trinity College. And she is one of, for me, the best researchers on ALS for a long period of time. And she has the ability to push the young people to study this devastating disease. We are working together in different European platforms. And today she's going to give us a talk, and the title is The Pathway to Precision Medicine in ALS. Thank you, Orla.
Gracias, Mónica. Lo siento que no puedo dar esta talk en español porque COVID. No puedo practicar mi español, so tengo que dar eso en inglés. So lo siento. So thank you very much for the invitation. It's a great pleasure to be here in this very historic city that actually has really good connections with Ireland. There's an Irish college here in Salamanca as well, which I'm hoping to visit after this meeting. So I'm going to talk actually very similar to my colleague Leonard Vandenberg's talk on the pathway to developing new treatments. And it aligns very well with the previous speaker just before lunch, about what we know, what we don't know, and what we don't know that we don't know. So these are my disclosures here. So just for people who may not be fully aware, so ALS is known as motor neuron disease, although clearly it's much more than that. We know it as a disorder of upper and lower motor neurons, and we know that it's a rare disease. The incidence is about 3 per 100,000 in populations of European origin, but probably much lower outside of Europe. It's more than a motor neuron disease. It's progressive, it's incurable, and it's terminal. And most people die of ALS within about 3 years of their first symptom. And because it takes about 12 months to diagnose ALS, we have about 2 years where we take care of patients before about 70% of them pass away. Now, we know that some cases are genetic, and they're associated with 4 main genes in the European population: CNNR72, TDP43, SOD1, and FUS. And Leonard is going to talk a bit more about the genetic component. We recognise ALS because it was described originally by Charcot, and it was described as amyotrophic, as in wasting muscles, lateral sclerosis, or hardening of the corticospinal tracts. And clinically, we are trained by the descendants of Charcot to recognise the upper motor neuron signs as weakness, brisk deep tendon reflexes, so loss of inhibition, and hypertonia, again a loss of normal modulation and muscle tone. And lower motor neuron signs, a loss of the function of the anterior horn cell with the signs that I described there. But of course it's much more than that, and one of the things that we've learned in the last maybe 10 or 15 years as we've studied this disease to a greater extent is the extramotor component to ALS. So we know that the motor system, the corticospinal system, in this very simplified diagram is involved. Of course, the corticospinal system is much more complicated than this diagram would have you believe. But there are also other parts of the brain that we know are affected. For example, the orbitus frontal circuit, which regulates emotion and behaviour, the dorsolateral prefrontal circuit, which regulates executive function, and the anterior cingulate circuit, which regulates motivation and initiation. And these aspects of the neurological system are also affected to greater or lesser extent in ALS. So it's not just a pure motor system. We've been studying this for many years, and we've done a number of population-based studies looking at the frequency of cognitive change in ALS, and we found that about 50% of people have some degree of cognitive change, maybe 60% or 70% have some behavioural change. And these are characterised not so much by the amnestic form that we see in the later Alzheimer-type disorders, but more an executive impairment, so an alignment with the frontotemporal dementias. And we, of course, know now that the two diseases are probably two ends of a neuropathological or clinical pathological spectrum. We also know there's significant language involvement in ALS as well. So if we look at those who are cognitively intact, they tend to remain cognitively intact. So already we can see that there's more than one clinical phenotype that links with ALS. We also looked at behaviour, and there are a number of different behaviours that we see, roughly reflecting the different circuits that I just mentioned within the frontostriatal and cingulatoris networks. So we see changes in initiation apathy. It counts for 60 to 70% of behavioural change in ALS, something we didn't really appreciate up to maybe 10 years ago. And we now have an apathy scale, a failure to adhere to social norms, difficulty with social engagement, interpersonal engagement, and difficulty with self-regulation. And these are present and can decline over time. These are difficult to measure, though, because they involve us making assessments of patients who come to our clinic or asking their relatives to tell us what they think about whether the person's behaviour has changed. So it's very difficult to measure these types of changes, for example, in a clinical trial setting, because they're very, very subjective. So these are the subphenotypes as they would link to the different pathways that I mentioned in the brain that we know to be affected in ALS. Now, how do you detect cognitive and behavioural changes? As clinicians, this is important. And the reason why it's important, because we need to be able to measure this in a clinical trial setting. So it's one of the aspects of ALS that we hadn't traditionally been measuring. And it's the other component, I suppose, of the disease, in addition to what we call the upper and the lower component, the cognitive component. So over the last 10 years or so, my colleague and Leonard's colleague Sharon Abrahams in Edinburgh, along with Thomas Back, has developed a screening tool called the Edinburgh Cognitive and Behavioural Screen, or the ECAS. And this is an example of the ECAS. So it's using what we know about the changes that occur at a cognitive level in ALS from the larger population-based studies that we've done, and then developing a short 15 or 20-minute screening tool that focuses on language, executive function, and then you also measures the non-ALS features to ensure that we're not missing other aspects of the disease, but also to allow us to characterise the changes to be specific to ALS relating to the executive function, the executive impairment. So this is just an example of the ECAS. How do we measure progression? Well, in ALS we use a scale called the ALS Functional Rating Scale, or the ALS FRS-OR. And it's a 48-point scale in 4 different domains. And actually, my colleague Leonard is going to speak next, is just back from a conference in Montreal. I was invited as well, actually, but I didn't go. But we know that there are problems with the ALS-FRS, but we use this scale in clinical trials, and we divide ALS into impairment of bulbar function, so speech and swallowing, impairment of fine motor, gross motor, and respiratory, and it's a 48-point scale, which is demonstrated here. And we also measure respiratory change, and we use these as the primary outcome measures in our clinical trials. And I'm going to come back to that a little bit later, because there are some difficulties with this, and this is where at some extent to some extent at the limit of the ability of this scale to deliver what we'd like it to deliver going forward in our future trials. One of the problems with the ALS-FRS is that it's not uniform. So ALS is a relatively heterogeneous disease. I mentioned earlier that some patients have cognitive change and some patients don't. Those who have no cognitive change tend not to develop cognitive change over time. Those who do tend to progress. So the variability in symptoms and severity, some patients have cognitive change, some patients don't. The site of onset, there's variability there. The rate of decline, as I've demonstrated there, the rate of decline is enormously heterogeneous. We have some patients who decline really rapidly and some patients who barely decline at all. And of course that also leads to changes in the likelihood of survival. We also know that the presence of cognitive change, particularly executive dysfunction, is a marker for the rate of progression. So if a patient presents with both evidence of cognitive change, particularly with respect to their verbal fluency, those patients are more likely to progress more rapidly than those patients who have no abnormality in cognitive function. You can see that the difference in the rate of progression in this slide here. Those with ALS with concomitant frontotemporal dementia, about 12% of people in our population already present with dementia. They progress much more rapidly than those who have no cognitive abnormality at all. So the rate is just under half in terms of the disease progression, indicating that the burden of disease and the presence of that cognitive domain is an indicator of the severity and burden of disease and the likelihood of progression. These are just graphs to show that. We know that there's a heterogeneity in survival, and these are studies we did many years ago looking at the survival rate between bulbar and spinal onset, and there are two graphs that are superimposed on one another over the 10-year period. Unfortunately, no change really in survival in our population-based study. But I want to show you is that about 70% of people die within the first 36 months. And then the other 30%, it's very difficult to prognosticate for those patients because they have a much more uncertain and often slower disease course. And we know some of the factors that determine that. The age of onset is important. Older people have a more rapid progression. The presence of cognitive behavioural change is important. The site of onset, bulbar, patients who present with speech and swallow trouble do worse than patients who present with limb trouble. The rate of progression already, so if the disease is progressing rapidly, it will continue to progress rapidly. And the genetic background, and I think Leonard is going to speak a bit more about that. We did some clustering a number of years ago where we were able to divide people into 3 categories, and the determinants of those were, as I shown, the presence of executive change. So those with the slowest form of decline were the ones who are most likely to be cognitively intact. So that may be a different subcategory of disease. And I think the point I'm making here is that ALS is a heterogeneous disease, and even very, very simple and somewhat crude assessments such as we can make in clinic can allow us to segregate patients out into different subgroups, so one of the determinants being the presence of our absence of cognitive change. Now, using just these clinical parameters that we have, including age of onset, site of onset, the presence or absence of dementia, and the presence of or absence of respiratory change, Leonard's group, using population-based data sets, developed this prediction model which predicts survival, and that's a very useful model, and that's a model that we are using in the enrolment of some clinical trials now going forward, the investigator-led studies. So that's a very useful tool that we have now, using these multiple 7 different components that we can mathematically develop an algorithm that will allow us to predict patients depending on whether the probability of survival is high or low for the course of the study. So this is very important in our ability to develop and modify and refine some of the clinical trial designs that we have. Now, one of the things that we referencing the talk this morning, one of the things that we think will help us to develop new drugs for ALS is based around the idea that if we found the cause, but actually it's going to be the causes of ALS, that will help us to develop drugs that will then help us to treat the disease. And I've been in the field for many years, and the field has evolved, and in a way it's evolved in a way not unlike the Alzheimer's story, where we probably know less about more. So we know that some of the very clear things that we thought were definitive, the sands have shifted considerably, and things that we thought we knew we don't know so well anymore. So one of the important points, and these are a number of caveats to the rest of my talk, is that the biology of the disease and even the crude clinical assessments that we can do as clinicians indicates that this is more than one disease. So I would like to think that ALS is a syndrome. It's not a single disease. It's like pneumonia or cancer. There are lots of different subtypes, most likely with different pathobiological causes and possibly different pathobiological processes that are driving progression. So it's not a singular condition, and that's really important because we need to be better at defining the subcategories of disease so that we can develop or drive better clinical trials. The risk for developing ALS is most likely different from the factors that drive disease progression. And at a talk last month, one of our colleagues, Jan Velding, made this sort of aphorism that, you don't treat lung cancer by stopping smoking. So identifying the causes of ALS aren't necessarily always going to help us to drive new treatments. It might help us to unpick the pathobiological processes. So understanding the factors that lead to ALS may well be different from how we might want to target that inexorable progression of the disease once it happens. And that's really important, because then we have to establish when does the disease start and who are those who are at risk. So it's very similar to the discussions this morning around Alzheimer's disease. Not all familial ALS is the same, and we know this even of the 30 genes, and Leonard will speak more about these. They have different pathobiological mechanisms as well, so the genetic causes, even the genetic causes, are not the same, and that's really important as well. And there have been some positive outcomes in recent trials and some not so positive outcomes in recent trials that reinforce that view. So, and finally, the concept of disease onset is one that we define as clinicians, and my view is that we were trained very well as clinicians by the descendants of Charcot, and our understanding of the biology is informed by the great histologist Raymond E. Cajal. But I think we need to be able to move beyond our clinical prowess and these clinical pathological correlations, fixed anatomy, into a much more dynamic view, so that we can understand what the onset really means of the disease, and whether the factors that predispose to disease, whether we can discern those or ascertain those at all. And then finally, we don't understand the underlying biological processes leading to clinical presentation. This is a theory, this is a hypothesis. We know that ALS is probably a gene-environment interaction, and it probably happens in 6 steps. And Amar Al-Shalabi from London, using population-based data sets, including ours, showed that ALS occurs mathematically in 6 steps, using some of the methodologies that are used in cancer, one of those being genetics, and that may be different for different populations, and also then environmental exposures, we don't know what they are, and then something else, time, aging, and then something else, and then the disease happens. We know that if you have a genetic variant such as SOD1 or C9orf, the number of steps declines, but not to 2. There are still 2 or 3 steps, fewer steps in SOD1, more steps in C9orf72. We know that ALS has a heritable component. Using our heritability estimates for our population-based registers, we can show that the heritability, the proportion of ALS risk that's determined by genetics, is about 50%. If you strip out those with a single gene and major effect, such as C9orf72, it's about 37%. But within C9orf72, there's probably a polygenic component to that as well. And these are the genes that we know are associated with ALS. Like I said, these 4 genes, or these 3 genes, are the ones that are targetable at the moment, and Leonard's going to talk about those after myself. But understanding the genes has allowed us to develop concepts modelling the disease in animals, and we have many different pathobiological mechanisms now based on the modelling that has taken place. And modelling is really important as long as we understand the limitations of the model. So we know that there are many, many processes that become involved in ALS. The relative importance of those processes may differ in different subpopulations, and that's something we don't really understand very well at the moment. Everything from impaired DNA damage repair, such as CNR72, right through to glial dysfunction and hyperexcitability, all aspects of the cell machinery in different iterations. We don't understand the biology of the cell as well as we should in this regard, and I think that was very clearly reflected this morning as well. What we have done is we've developed models. Models have been very useful. The model for SOD1, the best first model for ALS was the SOD1 mouse, the transgenic mouse developed in 1994. And many, many drugs were developed based on what we understood to be the pathogenic mechanisms involved with the SOD1 mouse. And these are all the drugs that were developed over the years into which we put clinical trials. And unfortunately, many of those drugs, all of the drugs that I've listed there, failed in clinical trials. So there is this valley of death from the preclinical to the clinical. And I think that, to my mind, there are two levels of responsibility here. There's a responsibility clearly from the preclinical end to understand the models and the limitations of the models. Very important to animal work, very important to understand the limitations. But we have a responsibility as clinicians as well at the other side. And the rest of my talk is about the responsibility that we have as clinicians in this regard. So the mouse problems with the anatomy is different. The genetic background is all the animal modelling was done on inbred strains of mice. There are genetic gender differences in the mice that nobody paid any attention to, and of course copy number differences as well. But also in the SOD1 mouse, the pathognomonic neuropathological fingerprint of ALS in humans is TDP43 deposition, and the SOD1 mouse and the SOD human does not have TDP43 deposition, so it's probably a different disease. We also know that the trajectory of the disease in mice is quite different. It's predictable, whereas in humans it's not at all, as I mentioned before. So what do? We should take a precision medicine-based approach to ALS. So that means the responsibility that we have as clinicians we need to take seriously. It's not okay to say, oh, well, we'd like to do a clinical trial on this drug because it's kind of interesting and there's a kind of interesting pathway. We need to be much better at and responsible and how we develop our clinical trials going forward. These are people that we're seeing, these are patients, but it's also about ensuring that we get best value for money for the investment that we make in our clinical trials. So if we use this precision medicine approach, I would contend that it will allow us to segregate patients into different subgroups and make our clinical trials more efficient and better value for money. So Leonard and I and Monica and Anna here from Anna Calvo here in Spain, we have worked together in the TRICALS, the treatment consortium to cure ALS, which Leonard leads and I helped lead. And we've developed this roadmap, this highway to a cure, which tries to address these post-translation or early clinical trial problems that we've had that have led to this failure of clinical trials, and much is to do with our lack of understanding of the disease, of course. But the contribution that we need to make to this is quite significant, and we haven't, to my mind, been paying as much attention to this as we should have. And we have the tools and technology to do that now. Our mission is to develop the right drug for the right patient at the right time and the right dose. And in order to do that, we put together this consortium, an academic industry collaboration called Precision ALS, which Leonard and I and Monica and others are leading. It's co-funded by the Irish state as well, and it's a collaboration between the TRICALS sites in Europe and ourselves and data scientists within our university and within the industry. And the idea is that we would collect everything that we can about patients, and we platform this in a dynamic machine and a dynamic platform with the data scientists, so that we can not only learn everything that we can about the patient, but continue to learn, to continue to refine. Use AI, use machine learning, but use it wisely and advisedly, and use as much technology as we can, as many of the biomarkers have been developed, to try and build this process whereby we can have these precision medicine-based clinical trials, or sometimes maybe looking at attributes of the disease rather than survival. So there's quite a lot of work that we need to do here. This is a 5-year project. How will this help us to navigate through the valley of death? Well, the failure of trials up to now have been, one of them is the drugs don't work, and that may be the case. The drugs may only work in some patients. We didn't have the right target engagement. We didn't know if the drug even got to where it should be. We might have used the wrong dose. many of the trials were poorly designed, and Leonard's group, he and Ruben van Eyck and the group in Utrecht have been pioneering the new concepts around clinical trial design and better outcome measures, better and more sensitive outcome measures. So what we learned from these is that we need better drugs, better trial design, better patient selection, better outcome measures, and better biomarkers. And I'm going to just talk about a couple of these. The landscape for clinical trials is very rich. There are a lot of new drugs coming down the market, and this is a slide that Chris McDermott from Sheffield has lent me, and it's just listed in the different pathogenic mechanisms. So a lot of activity going on, and we're wiser now than we were in the previous trials that we ran. And then there's also a lot of true precision-based therapies, and I'm not going to really dwell on these slides because I think Leonard is going to speak mostly about these. But the point is, it's a very rich field now. And we also need to be more responsible in the outcome measures. I mentioned that the ALS-FRS, we're asking a lot of that scale, and when we've dissected out the ALS-FRS, there are floors and ceilings in the ALS-FRS that really make it a relatively insensitive tool for the level of precision that we need now. It's the tool that we use. It's a tool that we'll probably continue to have to use because the regulatory authorities have learned to understand and interrogate this outcome measure, but we need better outcome measures. It is a limitation at the moment in how we do clinical trials. We also need better biomarkers, and there are a number of biomarkers already that are in progress. We use this idea of wet biomarkers, which are molecular protein cellular markers within that we can extract from patients. We can use imaging, MR and PET. We can use neurophysiological markers. We're going to talk a little bit more about that. And then we can use digital markers as well. And this, of course, COVID has really turbocharged our use of digital markers. We can think about biomarkers in a number of different categories. So biomarkers, we should define what we mean. So are they diagnostic? Are they prognostic? Are they markers of progression? Are they categorical? Are they predictive? And we can categorize each marker that we have, and we should, based on what we want the biomarker to do. It's not okay to say, we'll use this biomarker unless we say what we want the biomarker to do. And its robustness and its validity in the task that we set it to. Neurofilaments are biomarkers that have entered almost into the clinical domain now in both ALS, but in other neurodegenerations as well, and also in MS. And there is some evidence that these neurofilament-based biomarkers can be useful and can be predictive, and there are some data suggesting that if you lower these neurofilaments, that is indicative of a drug response. And this is the recently reported VALOR study for SOD1, where SOD1 was turned off using an antisense oligonucleotide, and the outcomes look very positive. So neurofilaments tracked with the likelihood of the drug actually working. What about novel and experimental biomarkers? And one of the things that as I've progressed in my career as a clinician scientist has been the frustration of the limitations of our own clinical prowess and the need to look at newer technologies that are beneficial, that can help us to learn things that we can't detect, even with the best and most advanced clinical training and clinical prowess that we have. So imaging is one. So imaging, we can image changes in ALS. This is my colleague Peter Bede, who works in my group, who's a neuroimaging researcher in ALS, and we can show white matter changes in ALS. And the other large imaging group in ALS in Europe is from the Netherlands, and the other one is in Italy. So there are a number of centres that are really interrogating the ALS brain and identifying both markers of disease and prediction, also being able to predict, and almost now getting to a point of single individual prediction. So and these markers have shown what we know clinically as well from our neuropsychology, that ALS is not just a disease of the motor system, that the basal ganglia is heavily involved as well. The caudate, the nucleus accumbens, and also some hippocampal involvement as well. And we can separate out the different subtypes of ALS. We can separate out C9 off signals of imaging compared to non-C9. But imaging has limitations. It's expensive. It's not suitable for patients who are progressing because they need to be able to lie flat in a scanner. The scanners have different computer-based algorithms, so it's difficult to undertake a study across many different sites. And it's very difficult. There are not that many comparable disease control data sets. So the other technologies that we've turned to, and I like the idea of network dysfunction. It's a direct measurement, doesn't interfere with metabolism. Functional MRI measures glucose uptake, PET scan measures ligand uptake. The excellent temporal resolution and the spatial resolution, which had a bad reputation, is now fixed with good computer modelling. We can now evaluate brain circuits, because actually ALS is probably a disorder of circuits in the same way as Alzheimer's is probably a disorder of synapses. So we can measure changes before cell loss, before we can see changes in structural MRI. We've published some work on this as well, and it's much cheaper, so you could incorporate it into clinical trials. And then we could do source analysis using modern mathematical technologies. We can identify the source of, if we undertake tasks, we can identify the source and demonstrate changes as well. So we can identify changes in resting state EEG using neurophysiology, and we can identify changes in task-related potential. So we can really start building a really strong positive platform, modelling of the brain using real-time electrical signaling analysis. And we can also start looking at clustering of patients using neurophysiologists. Paper we published in Brain recently, showing that patients with ALS can be segregated into different clusters, and those clusters are retained over time, and they're predictive of different subcategories of disease. For example, these clusters demonstrate different bulbar scores using the LSFRS, limb scores, respiratory scores, cognitive scores, behavioural scores, rates of progression and survival. So this is a new evolving technology that's being built. Our group and Leonard's group and others are driving this forward. It's a really exciting area. And I think this is one of the areas that's going to really help us to segregate the disease out. What are the next steps? Well, Leonard's going to talk about that to some extent, but Precision ALS will be the engine, in my opinion, in Europe that will drive this. So I think while we continue to really understand the disease at a basic science level, our responsibility as clinicians is to really extract as much information as we can from the patients using all the technologies that we have available, and use data analysis for that, applying all AI and machine learning to allow us to identify subgroups of patients using modern mathematical and computational tools. And this is what Precision ALS is about. So to conclude, ALS is a spectrum disorder. It's not a single disease entity. Multiple subcohorts exist with different pathogenic processes and disease trajectories. The early success with the SOD antisense oligonucleotides point to the future of genomic therapies, and data-driven approaches will help to tailor future treatments. And just one final pathway, there is some evidence now that there is a developmental component to ALS, and I suspect that we would also find that would also be the case in the other neurodegeneration as well. And the way you look at that is looking at endophenotypes and genomics in first degree and second degree relatives of those with these diseases. And we already have those type of studies underway. So to conclude, these are this is a team that I work with, and these are people who've generated most of the data today, and these are the funding sources. So thank you very much for listening to me.
Que no tenemos tiempo para preguntas ahora. Si acaso en el momento del café, during the coffee break, you can ask something to Orla. Thank you, Orla.